last talk before we go to the panel is by Dr. Ramille Shah, who delighted to have her here today. So she is co-founder and chief scientific officer at Dimension, but she started as an academic. So first in material science and engineering at Northwestern, and biomedical engineering at UIC, but is now full-time at her startup. So she has decades of experience in material development, particularly for 3D printing for, I mentioned, a variety of different biological applications, but not related to biomaterials, but interestingly also 3D printing even Martian, or the applications for metals and Martian and lunar dust, which is interesting. But yeah, so as I mentioned, started as an academic, but now transitioned full-time to try to translate and commercialize products. So great to have you.
It was, now it's on. Hello, well, it's a pleasure to be here today, and it's great to see actually faces I haven't seen in a while, so it's a great surprise. Thanks for the introduction and also the invitation to give a talk. It was a great introduction from Guillermo as far as the basis of how to commercialize, so I don't have to get into it. So it's going to be really me talking about my experience going out of academia and actually being part of the translation of our products. So it was, it's the best decision I've ever made because I've learned so much, and I really have found my, I guess, where I belong, I think. And it was always in sort of straddling the academics as well as industry, but really getting insights as far as how to actually take technology from the university down toward actual clinical translation has been a tremendous experience. And so hopefully I can share that with you and you can appreciate the difficulty, but also the reward at the end, and it's still not the end, so it's ongoing. So we've transitioned, we had many transitions along, and I'll go over sort of the pathway of Dimension Inc.'s, but we were first founded, I would say, in 2016, but we really started becoming a company when I onboarded my current CEO, Carolyn Collins. That's when we started to think about, before we just used our 3D printable biomaterials and we sold it to academic groups for them to use in their research, and that wasn't really getting us anywhere. So when we brought Carolyn along, we decided to really focus on creating products from the technology. And that's where we are now, where we started with our first product, which I'll go into, but then we transitioned to more complex products. But right now we stand as a regenerative therapeutics company. And so we took our first product to humans, which was exciting. And our platform is really one that really spans a whole variety of indications, from bone, which is our first indication, all the way to transition to ovary, transition to cell therapy, and now into liver disease cell therapies. But our journey has taken us where we are, and we're not here unless we were able to, have funding. So right now we've raised over $20 million in investments. These are all through VC, and we are having another round coming up. So we are excited about that as well. Although in this environment it's going to be kind of more difficult. But to get more into sort of the technology and our basis for the for what we've produced when I was at Northwestern, I got into using 3D printing in 2009, 2010, when it really was 3D printing was at its infancy as far as being used as a tool for tissue engineering applications. And one of the biggest things that we realized is that back then, there wasn't really many biomaterials that were both compatible with 3D printing, but also were tailorable to be able to address specific indications. So we wanted to create these biomaterial platforms that can be compatible with 3D printing. And what we discovered is one of the platforms that we've created was one which these inks are, these formulations are ones that basically allows us to print any material that comes in particle form. And so one of the first particles we printed was hydroxyapatite, which is the main mineral component of natural bone. And you'll see it here in this picture, SEM, where this is the 3D printed structure, but if you zoom in further and further, you have the fibers, then you have this texture that you see, but in the fiber itself, you can see the hydroxyapatite particles. And the ability to really control the composition and architecture among different scales, across different scales, has been powerful because we're able to then make tweaks to the design to be able to create specific bioactive behaviors when you start to interact with cells. So in this case, the composition, hydroxyapatite is for bone, but we can change it up with extracellular matrix particles, we can change it up with salt particles that then get removed and you leave behind a high porosity. So there's a lot of leverage we can use with regard to, with this platform to be able to tune the biological, the mechanical, and degradation properties. And this is important because not only do you want to control the cell behavior at the nano and micro scale, but when it comes to implanting devices, the host response is huge. So the ability to really induce the infiltration you need and the vascularization that you need from the host to be able to maintain the viability of the device is also something that we really haven't thought about in conjunction with cells on a material. Like, what is the response if you put cells in the material in the body? Well, how can you control the host response? And so that's one of the things that we've been really focusing on as we put more and more of our materials in vivo models. And so we're starting to pick out nice trends as far as how architecture plays a role, how composition plays a role, and how degradation plays a role. And this is just another image of what our materials look like. This is the one that was the we call this fluffy PLG, but this is our fluffy material. This is a 95+% porosity, so it feels very flexible. It's like styrofoam. But when the role of porosity within the macropores, but also intrafiber porosity played a huge role in how the in vivo response resulted. So in some cases, when you have no intrafiber porosity, you get this inflammatory FBR, the fibrosis that happens and the encapsulation that happens. When we started to put porosity within the intrafibers, we have a response that's completely different. It started to regenerate tissue within the macropores that then get vascularized, and there wasn't this fibrous encapsulation that happened. So the role of porosity, not just the macro, but the micro and nano, is really important. As I mentioned, there's a few different parameters we tailor in order to make these more amenable to different indications, composition, architecture, and microstructure. And the other thing is, when it comes to differentiation, as Guillermo had mentioned, it is important. It's not just it's a new material or whatnot, but this, using 3D printing, especially at the time we launched it into a publication, it was in 2012 when we published on our hyperelastic bone material. One of the things is we're able to use 3D printing in a rapid manner. So this is an extrusion-based 3D printing method. It's all room temperature. You don't need any heat or lasers. And so it's a rapid manufacturing that is amenable to scalability. The other thing is that the nature of the microstructure and the design allows us to have a variety of different materials that have different mechanical properties. So some things that are very flexible, as you see here, to something that's more rigid, but still can be cut very easily. And that's something, the surgical handling aspect is huge when it comes to the medical devices. And so being able to have structures that the surgeons can actually handle, it doesn't crumble on them, especially in the bone space, as Guillermo mentioned, it's very, very important. And then when we started to put these materials in the body, as I mentioned, we started to see this vascularization and tissue formation that we haven't seen with other types of 3D printed materials. So our first product was CM Flex. CM Flex was a first FDA cleared, fully synthetic bone graft, and it was indicated for bone defects in the face. So it's mandibular, maxillary. The indications include bone formation in tooth extraction sites, orthognathic cases where if the surgeon wants to actually move the jawline forward, you have these big gaps that need to be filled. So that's the first indication that we focused on. And CMFlex was really, strategic-wise, a product that we wanted to get on the market because it's the lower hanging fruit compared to going into complete organ regeneration or to cell therapies. We wanted to be able to create a product that gets through the FDA using our 3D printing materials and process. And so that was sort of the reasoning behind that. The other thing that it allowed us to do is actually build our quality management system for manufacturing to get into patients. And that eventually led to, of course, the first in human. I just wanted to take a little bit of time, though, just to give you an idea of how long it took us to get through 510(k) clearance. This is, again, the simpler device, a bone regenerative device. But we submitted our 510(k) in 2021, September. We went through several iterations with the FDA, but about 15 months later, we actually got 510(k) clearance. And the challenge was when we first got the under substantive review. This is when the FDA gives their actual evaluation of what other studies they want you to do. Because it was the first 3D printed material that they are clearing, they wanted to have some more safety and biocompatibility testing. And so we needed to do some more genotoxicity and systemic toxicity testing, which required animal models. So that took about 6 to 8 months to complete, and that's why it took about 15 months altogether. But at the end, I think it was definitely worth it. There was a point, honestly, where we thought we were going to give up because that was actually when we got this, the response. It was in the middle of COVID, right? The whole after effects of COVID, and how can we execute this in a timely manner without running out of money? And so that's the decisions we had to make, but we decided to move forward. So one of the things I think would be useful is to show the evolution of how we got from bone to where we are now. And so, as I mentioned, we started 2019. The next year, we did make the decision to start developing the CMFlex product, and that's where we raised our first seed round. We had in 2021, we figured what is our next product? And in the next product, we wanted to have something that's more not complicated, but something that involved cellular delivery within our materials. And that's when part of the work at Northwestern was this artificial ovary project that we had with Teresa Woodruff, where we, in this case, we focused on using our 3D printable materials as a temporary housing for follicles for patients who are undergoing radiation therapy, because usually the radiation therapy actually destroys the architecture of the ovary and they become sterile. And so the concept was actually taking the follicles from the patient before they get the treatment, putting it in our 3D printed scaffolds, and maturing those in vitro or implanting them in the patients to recover their fertility and hormone production. In this case, our first product we were thinking of is using our materials as an in vitro maturation device to augment what currently in vitro fertilization is doing, because the efficiency of in vitro fertilization nowadays is not as great, and so we can use our materials as environments to help enhance that process. But as you'll see, in 2023, we made a decision to terminate this program. And the reason why we decided, because when we were doing market analysis and adoption analysis, the infrastructure needed to actually adopt this type of product at the time was too much. It wasn't worth it. And so that's where I'll go into the lessons learned. But one of the things, even though it's a cool technology, we had the first this was a Nature Communications paper, we had the first showing that, if you put follicles within our device and implant that in a sterile mouse, it actually resulted in in pups being born. So it was a first sort of organ-like 3D printed structure that actually worked. But even though the science was cool, that doesn't mean that it can be easily adopted as a product. And all the complications that go with it, monetary, economically wise, wasn't worth it. And so we said, okay, well, how do we pivot? Interestingly, in 2022, we started to have these first conversations with partners that were interested in using our device and materials for cell delivery. And this is where creating cell therapy and using our materials to help the engraftment of cells that are delivered in type 1 diabetes came into play. That was in 2023, this is the nature of startups, this partner actually got acquired. And so that project got dropped. But we had great results from the T1D initial experiments. And so we decided to start to, see, is it something that we should go on our own? So we sort of put it on pause, but what we ended up doing is taking a shift and seeing where is there a need in cell therapy, and liver disease actually went to the top. And so in 2024, last year, we actually decided to now put all our focus on using our materials to help augment cell therapies when it comes to liver disease. I know the actual technology and why is not, I haven't said why, but the fact that our materials can actually enhance vascularization, we have shown that it can also enhance cellular engraftment for cells that are actually incorporated within our materials. So we've had great success so far. So we're really using the foundation, as I mentioned, as far as the bone work and getting first in clinical. So our first clinical case for the CM Flex product was in 2023, but that really showcased that our materials are actually safe and it can be put in humans. We can manufacture it at a scale and in a quality system that is regulated. We actually just got, we were successful in passing our first FDA inspection last month, which is a huge endeavor and accomplishment, because that's something that says that we're doing something right with regard to manufacturing. And so, as you see here, acellular going to autologous and then now into allogeneic cell transplantation. It gets more complicated, but we're actually here now. And woo, there you go. And so this is the last slide. I just wanted to share some in sort of not just focused on CM Flex, but in general, right? Because we've gone through a lot in the just past 5 years. But one of some key highlights, these are not all of it by no means, but one of the things that came to mind when it comes to various different aspects of translation, market research is very important, not just in the beginning, but also in the middle, but also at the end, and even before you launch. Because what we've found that things change over time, right? Of course, in the beginning, are you fulfilling a real need? And what is the differentiation? Can you actually get it adopted or get surgeons to use the product? Product form factor and size was a surprise, because we initially thought a specific size is what they wanted first in the CM Flex. This is a bigger block, but right before launch, people were asking, or surgeons were asking for smaller because of pricing. They're wasting a lot of product, and so that's something that was overlooked as far as what is the actual size that the majority of surgeons wanted. Product use and handling. So as I mentioned, porosity in this case is very important, because if you crush the product, the tissue will not integrate as well. And so the handling and how these surgeons, if you see these orthopedic surgeons or these surgeons who work with bone, they're very rough, I would say. But they're not gentle. Like, when we did our animal models, perfect cutout, put it in the animal is golden, like everything, it worked perfect, bone regeneration throughout. Come to surgeons in human cases, crushed it here and there, not has good integration. So there's a lot of things we started to learn when I actually got to the human cases. And performance expectations. So this is where the degradation rate of materials. What is the expectation of a dentist who wants to put an implant in within 6 months versus somebody who's doing an orthognathic case that wants regeneration of a full segment? one wants complete remodeling in 3 months, another can wait till six months or a year. So those are differences that you need to, as soon as possible, know so that you can know who to target and where your product fits the best. Cost and reimbursement is something that is very important, especially in the development process, because every decision you make in the manufacturing of your device increases costs. And is there, when it comes to the more exotic indication like cell therapy, are the insurance providers going to reimburse? And because those are a lot more expensive versus medical devices, that's an important part in our research today as we get into cell therapy. Infrastructure workflows. So this is where I mentioned the ovary. This is the main reason why we stopped that project. And the most important thing, one of the things, especially as the CSO, is what data will be convincing for surgeons in order for them to adopt. Do you need human cases? Can you get away with large animal models? So getting a sense of what their expectations are is huge, because that can impact your planning and fundraising as well. So that's another thing you should keep in mind. Regulatory manufacturing QC. We went the consultant route. We didn't hire anybody in-house, but choosing the right consultant, not just the cheapest one, but really somebody who actually knows the space is important versus a general one. I think that's one of the things where I mentioned that we started off with one consultant because they were cheaper, but if we had spent a little bit more money for one that was actually more in tune with this maxillofacial area, I think we could have gone faster. But it is okay to switch in the middle. The earlier you can start QC, which is quality control, the better, because the documentation needed is to really bring a product to market is huge amount. And this is just for CM Flex. Can you imagine for cell therapy, the quality control that's needed for both the material and the cells is a huge endeavor. So we're thinking about that now, even though we just started. And then, as I mentioned, the COGS. And lastly, Guillermo just said how important the team is. It is important to choose the right team from the very beginning. And if you make bad choices or somebody doesn't work out, try to fire them fast as you can. Because especially in a small team, a bad link makes a huge impact, and that could really slow down just the culture. I think one of the things we did right from the beginning is focus on culture, and that's something in academia I haven't really focused on, but in industry or trying to commercialize something, the importance of culture makes a huge difference as far as the turnover. You want people to stay, you want to keep good people, and so building a good culture is something that we keep at from the very beginning, and I'm proud of where we are now. The team changes fast. As you go through different cycles or different, from the beginning, middle, down to the clinical use, the team starts to change. What you need, what expertise you need. And so being able to foresee what you need before you actually need it is important, because planning, I think the biggest thing in my job, if I boil it down to what I do on an everyday basis, is gather information, make decisions, and try to foresee risk. And part of the risk is, who do we need in our team a year from now so we can hit our targets? And so that's something that I've had to get used to in the industry world. And then partners, the same thing. You think that, your internal team you can control, but if you partner with somebody that doesn't fit in your culture, it can be bad news as well. So that's something to keep in mind. And also who you partner with, if they can't move fast enough, they can slow you down as well. So that's something to take into consideration. Thank you. Appreciate Sean.
We have a couple minutes from here, just a couple questions.
The microphone. Hi, thank you for the talk. It was really nice, and I love the pipeline. Like you explained it really nicely. So, my question is like 3D printing was one of these technologies that I was sold. Like we will be able to print like functional hearts, right? And I think that a lot of investors and startups got excited about that. But the reality of the science is that that's not going to be possible. So, how do you manage investors' expectations with the reality of the scientific progress? So you tell them, okay, this is what we can accomplish, but then they don't get, they don't pull their money back, right? So I don't know if you have a problem with that or.
Yeah, so I think when you get to the stage where you're raising money in a tight environment like this, you need to, I don't know your plan, and it needs to make sense. And VCs, they know. They can sense when you're not prepared. So all the work that goes into getting ready for fundraising is a huge amount. Our leadership team, that's the function of our team here, we start 18 months beforehand. And that's planning, and in the science, in the, I guess, R&D side of things, what are the major milestones that we need to accomplish, and it's not just on the R&D side, it's also on the regulatory side. So we're getting ready to go into an Interact meeting with the FDA right now and getting prepared. So what are the major milestones on the R&D front, the regulatory front, the marketing front? You have to cover all your bases. So you have to tell them a story that seems reasonable. We always took a pragmatic approach. We're not saying that we can, build replacement livers. We're saying that we can use our materials to augment cell function and viability when they're implanted, right? So you have to, I think that we take them in steps. Sure, we'll learn through developing that product that we can potentially get to that point far in the future probably, but it's just too complicated, right? I think I think you have to be realistic and pragmatic and know your stuff. Mm-hmm.
So maybe we'll do, Joyce, one quick question.
Yeah.
Well, maybe if Chris and Diane can come on and walk.
Okay, yeah, so this is
their way
great, and I'm wondering how you disseminate your lessons learned. So I'm wondering I don't know if I've seen some people sometimes publish, in maybe societies or I don't know if there's journal articles, or what do you think if you had, I realize you're the first to do this, but what would you like to see, like in terms of like dissemination, whether it's journals or some other format that you would learn from or that you pass on knowledge, and also your failures? Yeah.
No, that's a great question. And that's something that, participating in things like this, I think that's why I thought it was a great opportunity. And there are also a lot of effort, not just locally, but nationally, groups that are trying to help accelerate translation of technologies. I've personally been involved in those to be able to, but most of my interaction has been through meetings like this, or talking in small groups, or being part of groups that are how can we improve and get it out. But I like your idea as far as, white papers, probably blogs, right, if I had time to do that, or maybe when I retire, we can, do what do you call it, podcast? But, there's so many different avenues now to get that word out. But I do think there are efforts in starting to think about those things, so it gets out there sooner than later would be great. Mm-hmm.